You've been there. It's 2 a.m., the textbook is glowing under the desk lamp, and you finally nail that complex equation. You feel completely confident. But then test day arrives 72 hours later, and the information just slips right through your fingers. We usually blame ourselves. We assume our memory is like a faulty hard drive, where data simply leaks out over time due to a passive glitch or lack of focus. But this memory loss is highly predictable. In the 1880s, a German psychologist named Hermann Ebbinghaus spent years testing his own memory to see exactly how fast new information degrades. This graph shows the result of his work, now known as the Ebbinghaus forgetting curve. Without active reinforcement, your retention of newly learned material plummets 50% within the very first hour. Within a day, 70% of it is gone. Losing the vast majority of what you learn within a single rotation of the Earth feels like a biological failure. It seems as though our brains are intentionally sabotaging our hard work. Modern neuroscience reveals that the leaky bucket theory of memory is an oversimplification. Recent discoveries indicate that forgetting is a deliberate, precision-engineered biological function. The brain utilizes specific cellular pathways to dismantle recently formed neural connections. Maintaining every single synapse would overwhelm our neural circuits. Consequently, the brain consumes significant metabolic energy to erase irrelevant data, clearing the path for new learning. To master your memory, you have to stop fighting what you perceive as a flaw. You have to understand this invisible architecture of erasure. New factual information, like the math formulas you're trying to memorize, begins to take physical form in a medial temporal lobe structure known as the hippocampus. When neurons fire together repeatedly during learning, they strengthen their physical connections with one another. Neuroscientists call this long-term potentiation, or LTP. This diagram illustrates the mechanism underlying LTP. Focus on the middle panel. The brain physically remodels the synapse by inserting new AMPA receptor proteins into the cell membrane, effectively locking in that high-frequency transmission. But the brain operates under strict constraints. It has a limited cognitive load capacity, and maintaining every single one of these newly potentiated synapses requires massive amounts of energy and physical space. To counter this, the brain triggers LTP decay. Look at the right panel. This active mechanism internalizes those AMPA receptors, pulling them back out of the synapse. The connection weakens and the memory fades. Your hard work is literally dismantled. The brain prunes its own connections to prevent network saturation, keeping your mind adaptable and preventing sensory overload. When Ebbinghaus mapped his forgetting curve in the 19th century, he was studying in relative quiet. He didn't have to contend with the relentless barrage of digital stimuli we face today. Engaging with multiple screens and notifications splits your attention. Without sustained focus, those initial memory traces are never properly encoded in the hippocampus to begin with. On top of that, the continuous rapid dopaminergic rewards delivered by short-form videos trigger structural hyperactivation in the prefrontal cortex. Over time, this intense stimulation erodes your executive functions. The working memory and inhibitory control needed to sit down and focus on complex academic tasks start to degrade. Researchers refer to this decline as digital dementia. While not a clinical diagnosis, it describes a severe, real-world drop in short-term memory and cognitive focus caused by excessive passive screen time. Trying to force formulas into your head is an exercise in frustration if your daily digital diet has compromised your brain's ability to maintain attention and initiate that critical long-term potentiation. So, how do you bypass this active deletion process and save the information you actually want to keep? The answer lies in the specific architecture of your sleep cycles. Both non-REM and REM sleep are vital for finalizing memory storage. While you sleep, the brain performs a transfer process called systems consolidation. Fragile, newly formed memories are reorganized and physically moved from the short-term storage of the hippocampus to the cortex, where they become permanently stable. Simultaneously, sleep performs global synaptic downscaling. It actively weakens the background noise of unimportant daily connections, which highlights the signal of the vital information that needs to be saved. Skipping sleep forces your brain to operate on fragile, unconsolidated data. These nocturnal hours provide the necessary metabolic window to build a stable, long-term architecture for your intelligence. To flatten that steep drop-off of the Ebbinghaus curve, you need a specific behavioral intervention, spaced active recall. Testing yourself repeatedly at gradually increasing intervals sends a biological distress signal. It forces the neural circuits to halt LTP decay and resynthesize the proteins needed to strengthen those fading synapses. To make this work, you must eliminate media multitasking during your study sessions. Protecting your prefrontal cortex allows you to achieve the deep focus necessary to trigger LTP in the first place. Memory operates like an active, energy-consuming muscle. It requires the focused stress of attention, the repetition of spaced recall, and the deep recovery of sleep to survive. So close the textbook, turn off the late-night lamp, and get some rest, letting your biology do the work it was designed